Welcome friends. Today we will solve few important questions on biochemistry. So let's start with number 1. A protein that shows infinite cooperative for binding of n ligands will be. So for infinite cooperativity, guys, what we have to remember that protein will only be found in either the unliganded form or the fully liganded form. Okay, not in intermediate form, but unliganded or fully liganded, or it will show a Hill coefficient of n. Okay, then it will show an infinite cooperativity. So both B and C point are correct. So D will be the correct option. Question number 2. If the oxygen is labeled in CO2, okay, this oxygen is labeled in CO2 and this CO2 has been provided to the plant. The labeled CO2 has been provided to the plant. Where is this CO2 will be expected to be seen after the plant has undergone photosynthesis. Okay, it is asking that where this labeled CO2 will be found after photosynthesis. So guys, the CO2, when it is given to the plant, it is incorporated into carbohydrate. Okay, so in carbohydrate, this CO2, labeled CO2 will be seen. Okay, number 3. In RNA, uracil pairs with. So let's know about DNA first. Then we will come back to RNA. In DNA, adenine base pairs with thymine. In RNA, in place of thymine, uracil is present. So adenine is pairing with uracil in RNA. So A will be the correct option. Question number 4. Question number 4 is a redox potential means substance has. So a redox potential, a positive redox potential means a substance has higher affinity for electron. Okay, when the redox potential is positive means the substance has higher affinity for electron. Okay, now number 5. How many molecules of ATPs are produced per NADH oxidation? So we all know that near about 3 ATPs are produced per NADH oxidation. Okay. Next question, number 6. Which trace mineral is a component of the enzyme that activates vitamin A in the eye? So the component of the enzyme which is activating vitamin A is zinc. Okay, so the enzyme which is activating vitamin A requires which mineral element? It is zinc. Next question is number 7. Which can be used as a mobile phase in HPLC application? So guys, for high performance liquid chromatography, any compound with solubility in liquid is used as a mobile phase. Any compound with solubility in liquid. Okay. Next, number 8. Infants are most susceptible to bacterial infection due to low circulating levels of IgG. So which age infants are susceptible to bacterial infection because they have low IgG? Okay, so answer is at the age of 3 to 12 months. Okay, these infants are very susceptible to bacterial infections. Okay, next number 9. If one cell has 80 content up to 40%, okay, what will be the percentage of guanine residue? Okay, so we know total of A plus T is equals to G plus C. So if A plus T is equals to 40%, then so we know that if AT is 40%, then GC will be 100 minus 40. Okay, GC will be 60%. So if GC is equals to 60%, that means 30% is guanine and 30% is cytosine. Okay, so here answer will be G is equals to 30% and C is equals to 30%. We are asked about guanine, so guanine will be 30%. Number 10. In sperm heads, DNA is particularly highly condensed and the histones are replaced with small basic protein. Okay, in sperm, the basic protein which is there is protamine. Okay, the basic protein which is present in sperm is protamine. This is very important, guys. Next is number 11. Number 11 is Taylor Woods and Hughes labeled VCR DNA by allowing new DNA synthesis in the presence of radioactive thymine. After DNA replication, S phase of the cell cycle, it was observed that. So here, after DNA replication of the cell cycle, in the S phase of the cell cycle, it was observed that both chromatids of the chromosome were labeled. Okay, it was observed that both chromatids of the chromosome was labeled. So next is number 12, affinity chromatography deals with. So affinity chromatography deals with specific binding of a protein constituents for another molecule. Okay, there will be a specific binding of a protein constituent. Okay, this is affinity chromatography. Number 13 is molar absorptivity is the measure of amount of light absorbed per unit concentration. Okay, it is the amount of light absorbed per unit of concentration is referred to as molar absorptivity. Okay, next question is number 14. It is restriction enzymes are named for. Okay, the restriction enzymes are named for, they are named for the bacterium they are derived from. Okay, as you remember the name EcoR1, so Eco is from E. coli, so they are named from the bacteria they come from. Okay, so answer is B. Number 15, both DNA gel electrophoresis and SDS page of proteins are similar because. Why the gel electrophoresis and SDS page are similar? Okay, why they are similar? In both cases, molecules migrate to the anode. Both techniques rely on a constant charge to mass ratio. Both techniques utilize the sieving properties of the gel. So all of the points are correct. That is, molecules migrate to the anode. There is the techniques they rely on is a constant charge to mass ratio, and they utilize the sieving properties of the gel. So all of the options are correct over here. Number 16, a person with phenylketonuria is advised not to consume which of the following product? Okay, so he is advised not to consume aspartame. Okay, as you remember, aspartame is an artificial sweetener. Okay, next is number 17. Question number 17. In a typical electrophoresis experiment, larger fragments of the DNA move more. So if you remember that in gel electrophoresis, the larger fragments move slowly, so they appear nearer the top of the gel photograph. You remember the larger fragments remain towards the top and the smaller fragments move downwards. Okay, A is the correct option. Next, 18 number, which of these vitamin is a vitamin A precursor? So vitamin A precursor is, next is number 18, which of these is a vitamin A precursor? So remember, vitamin A precursor is beta carotene. Next is DNA with a GC content of 50% will melt at approximately. So when a DNA with a GC content of 50%, it will melt at approximately 70 degrees centigrade. Okay. Number 20, the lone pair of electrons on oxygen in a water molecule. So these lone pair of electrons in a water molecule, they carry a partial negative charge. Okay, lone pair carry a partial negative charge. Now, next question is, amino acids required in human diet are not synthesized by the body are called. So these amino acids which are not synthesized by the body are called essential amino acids. Okay. Next is, the alpha amino acids have a carboxyl group with pK around dash and an amino group with pK near dash. So we have to select the best options in the given. Okay, so answer is the alpha amino acid have a carboxyl group with pK around 6.5 and the amino group with pK around 8. Okay, next number 3. The correlation between free energy del G transfer between the aqueous and organic phases and the surface area of amino acid residue. It is asking us the, just a moment guys, the correlation between free energy del G transfer between the aqueous organic phases and the surface area of amino acid. Okay, so what will be the correct answer? Correct answer is the correlation reflects the reduction. It reflects the reduction in solvent accessible area during protein folding. Okay, it reflects the solvent accessible area during protein folding. Number 4, lymphocytes are activated by antigen in the. So lymphocytes are activated in the lymph node. Okay. Question number 5, IgE provides. So immunoglobulin E, it provides immunity against certain parasites. Okay, it's mainly responsible for allergic reactions, so it provides immunity against some parasites. Next is, which one of the following is correct? Membranes from boundaries around the cell and distinct subcellular components. Membranes act as selectively permeable barrier. Membranes contain varying amount of lipid and protein and some contain small amount of carbohydrate. So this is correct. All of the statements seem correct. Membrane, they form boundaries around the cell. They act as selectively permeable membrane, also called to be a semi-permeable membrane. They contain varying amount of lipid and protein and some contain small amount of carbohydrate. So all of the statements are correct. Okay. Next is enhancers are the region. So what do enhancers do? Okay, enhancers are the region. They modulate transcription. Okay, they are present upstream or downstream of the transcription start site and they modulate the transcription. Number 8, transcription of each set of rRNA genes by RNA polymerase 1 produces. Okay, transcription of each rRNA genes by RNA polymerase 1 produces. So it produces a single 45S rRNA precursor molecule. Okay, it produces a single 45S rRNA precursor molecule. Next is, which of these restriction enzymes produces blunt end? Okay, so most of the restriction enzyme produces sticky end. The one producing blunt end are very few. Among those, it's EcoRV. Okay, EcoRV produces blunt end. Next is, the production or breakdown of dash is often coupled with the metabolic reactions of biosynthesis and catabolism. Okay, the production or breakdown of dash, so the production and breakdown of ATP is often coupled with the metabolic reactions of biosynthesis and catabolism. Okay. Next is, cellulose fibers resemble with the protein structure in the form of. So cellulose fibers resemble with the protein structure in the form of beta sheet. Number 12, the ultimate source of energy that sustains living organism is. So ultimate source of energy is number C, which is sunlight. Next is the number of water molecules consumed by the formation of phosphatidylserine. So for the formation of phosphatidylserine, which is a component of the inner membrane, the number of water molecule consumed is 0. No water molecule are consumed in the formation of phosphatidylserine. Okay. Number 14 is long chain fatty acids are oxidized stepwise in 1 carbon units. starting from the, so where are they started? Okay, so they are starting from the carboxyl end, long chain fatty acids are oxidized. Okay, remember, starting from the carboxyl end. Next is, as a result of photosynthetic process, which product is formed? So as a result of photosynthesis process, oxygen is the byproduct which is formed. Number 16 is, if the oxygen is labeled in CO2 and provide this CO2 to the plant, where it is expected to find this labeled oxygen after the plant has undergone photosynthesis. So this question we have already done, that when the CO2 is labeled after the photosynthesis, this CO2 is, the labeled CO2 will be seen in carbohydrate. Okay, this is a repeat question. Then question number 17, chronic consumption of excess selenium can lead to. So when the selenium will be taken in excess, it causes muscle weakness. Next, what does Michelson interferometer do? So what is the role of Michelson interferometer? So the role is, it modulates the IR signal at lower frequency so that it can be observed by a detector. Okay, it is modulating the IR signal at lower frequency. Okay, next, number 19. The enzyme responsible for breaking the hydrogen bond and thus separating the DNA strands during DNA synthesis is. So the enzyme which is separating the DNA strand by breaking the hydrogen bond, it is helicase. Okay. Next is an enzyme that recognizes foreign DNA and results in a cut in that DNA is. Okay, so this we have discussed in detail. It will be restriction endonuclease, which will recognize the foreign DNA and will cut that DNA. That will be restriction endonuclease. Now, next question is number 1. In beta plated sheet structures neighboring, okay, in beta plated sheet structures neighboring, so what will be the correct option? It will be neighboring chains are hydrogen bonded. Okay, so the neighboring chains are hydrogen bonded in the beta sheet. Number 2, bisphosphoglycerate BPG cannot bind to the oxygenated R state of hemoglobin because. Why BPG cannot bind? Because it is displaced from the, because its binding pocket becomes too small to accommodate BPG. Okay, so because the binding pocket, its binding pocket becomes too small to accommodate BPG. So this is the correct option. The nucleophile in serine protease is. So the nucleophile in serine protease is both serine and water. Okay. Next is, what would happen if serum is omitted from the ELISA but all other steps remain same? So if serum is omitted, anti-human Ig conjugate would not bind and will be washed away. Okay, anti-human immunoglobulin conjugate would not bind and it will be washed away, and the OD values would be nearly the same as the assay control. Okay, so both the options are correct. Answer will be number C. Number 5 is, an antigen preparation and an antibody preparation are tested by immunodiffusion. 3 bands are found indicating that. So what does the 3 bands indicate? So 3 bands indicate that there was more than one antigen. Okay, there was more than one antigen. Next, bacteriorhodopsin. Bacteriorhodopsin, what is this? It absorbs light and pumps proton. It is an integral membrane protein. It contains primary alpha helical residues. So, if you have seen the signaling pathway of bacteriorhodopsin, uh, you must be remembering that it absorbs light and pumps proton, and it is an integral membrane protein and contains primarily alpha helical residues. So all of the options are correct here. Answer will be number D. Number 7. Cholesterol is an essential for normal membrane function because. So why cholesterol is essential? Because it spans the thickness of the bilayer. I'm very sorry. Because it keeps the membrane fluid. That is why cholesterol is essential. Okay. Question number 8. In terms of cell communication, what do bacterial pathogens such as cholera and anthrax have in common? So, in terms of a cell communication, what do bacterial pathogens like cholera and anthrax have in common? So both of them, they can block the normal functioning of signal transduction process. Okay, both cholera and anthrax, they have the capability to block the normal functioning of the signal transduction mechanism. Okay. Number 9, which one of the following is incorrect? Okay, we have to find the incorrect one. In the DNA double helix, two strands of the DNA are bound with each other with the bases. Next is adenine always pairs with thymine, guanine always pair with cytosine, and none of the above. So, which one of the following is incorrect? So, here is in DNA double helix, two strands of the DNA are bound with each other with the bases. Okay, here it should be with the name of the bond should be given. Okay, now adenine always pairs with thymine, so there are exceptions whenever the word always is given. These kind of statements are most of the time incorrect because exceptions always exist. So here none of the above is correct. Okay, next is in RNA, uracil base pairs with this we have already done. It base pairs with adenine. Next is the binding of lac repressor to DNA could be considered to be analogous to. Okay, so binding of lac repressor would be considered to be analogous to competitive inhibitor of an enzyme. Okay, it is analogous to competitive inhibition of an enzyme. Next is number 12. Fructose is metabolized by. So fructose is metabolized by fructose 1 phosphate and fructose 6 phosphate pathway. Okay, so both A and B are correct. Next is where the acyl CoA formed in the cytosol is transported for oxidation. So this acyl CoA is transported to mitochondrial matrix for oxidation because this is the region where oxidation will take place. In what form does the product of glycolysis enter the TCA cycle? So we all remember that it enters in the form of acetyl CoA. Okay. Next is NADP plus is reduced to NADPH during. So in the light dependent reaction of photosynthesis, NADP plus is reduced to NADPH. Okay, next question is number 16. The frequency of precession, the transition frequency and the Larmor frequency are. Okay, so they are the different terms for the same frequency. So the frequency of precession, which is the transition frequency, and the Larmor frequency, they are the different terms with the same frequency. Okay, next, doubling the column's length increases resolution by a factor of. So when the column's length is increased, when the column's length is doubled, then resolution is increased by a factor of 2 to the power 0.5. Okay. Next question number 18. Next is number 18. Column efficiency is measured in terms of number of plates, which is. So column efficiency is measured in number of terms, in terms of number of plates, which is inversely related to the square of the peak width. Okay, column efficiency is inversely related to the square of the peak width. Number 19, number of times the two DNA strands are intertwined in a circular DNA is known as. Okay, it is known as linking number, which is the number of times the DNA strands are intertwined in a circular DNA. Okay. Number 20, the function of gluconeogenesis is. So, maintenance of blood glucose levels during starvation or low carbohydrate diet, recovery of lactate from fermentation and glycerol from fat breakdown, a pathway for the utilization of amino acids, all of the above. So, all of the above is the role of gluconeogenesis. Okay. Now, so let's start again. Which of the following structure is expected in a bacterium? So, which of the following structure is expected in a bacterium? So, this will be plasma membrane. Okay, in a bacterium, we cannot expect the organelles to be present, and the nucleus is also not true nucleus, so we can expect the plasma membrane is present. Next is the nature of peptide bond can be best explained as. So the nature of peptide bond will be best explained as the partial double bond. Okay, this question is very important, guys. It is repeated most of the time. Number 3, which one of the following is most correct? Okay, let's read. Charged amino acids are never buried in the interior of the protein. Next is charged amino acids are seldom buried in the interior of a protein. All hydrophobic amino acids are buried when a protein folds. Tyrosine is only found in the interior of the protein. So answer is charged amino acids are seldom buried in the interior of the protein. Okay, they are not exposed. Next is a pathogen can be a. So a pathogen can be an agent that causes the disease. It can be virus, it can be bacteria, so it can be all of these. Okay, next. Number 5. Which of the Rous sarcoma virus has a homologous cellular protein? So, CSRC is a Rous sarcoma virus which has a homologous cellular protein. Okay, next is number 6. If one cell has 80 content 40%, what will be the percentage of guanine residue? Now, this question is also repeated. The answer will be number C, which is 30%. I have already explained this in a previous video. Next, number 7 is methylated genes are. So methylated genes are. So friends, this you have studied a lot of time. Just guess the answer, then I will tell you. So these methylated genes are silent genes. Okay, when they are methylated, they become silent. Next is, why does the glycolytic pathway continue in the direction of glucose catabolism? Okay, so there are essentially three irreversible reactions that act as a driving force for the pathway. So this is the reason why glycolytic pathway continue in the direction of glucose catabolism, because there are three irreversible reactions that are acting as a driving force for the pathway. Number 9 is ATP is from which general category of molecules? Okay, so ATP is from the nucleotide category. Okay. Next is number 10. The key enzyme in the regulation of fatty acid synthesis is. So for fatty acid synthesis, the key enzyme is protein phosphatase. Okay, the key enzyme is protein phosphatase. So guys, please look at the chart. So I recommend when you are solving questions with me in this video, I recommend that please have at least little preparation in this unit, in the unit of biochemistry. Then it will be easier for you to solve the questions, because this video I have made for you to practice. Okay, we are not going into detail, we are just revising the concept. Okay, how, what kind of questions which are common and which comes a lot. Okay, so that I have, for that I have made this video. Okay, so continue. Fatty acid synthesis takes place in, so where does the fatty acid synthesis takes place? Now this question is again very important, it comes a lot. Okay, where do the fatty acid synthesis takes place? So it takes place in mitochondria. You are perfectly correct. It takes place in mitochondria. What we are talking about? We are talking about fatty acid synthesis. Next is cholesterol is the precursor of, now this is question is also very important. Cholesterol is the precursor of steroid hormones and bile salts. Okay. Vitamin A precursor we have already studied. The precursor of vitamin A is beta carotene. Okay, for steroid and bile salts, cholesterol is the precursor. Next, number 13, a fat soluble vitamin that regulates blood clotting is. So the vitamin which regulates blood clotting is vitamin K. Now vitamin K was first isolated from alpha alpha and has phytyl side chain consisting of. Okay, the phytyl side chain of vitamin K consists of 4 isoprene units. Okay, if you remember, I have taught you in secondary metabolite that one isoprene units consists of 5 carbons. Okay, it has 4 isoprene units. Number 15, which of the following mineral deficiency may result into impaired growth and development, skin lesions and loss of appetite? Okay, so all these are due to the deficiency of zinc. Okay, if you remember, we have already done in previous videos of practice MCQs that zinc is the mineral ion which is required uh, for the enzyme which synthesizes vitamin A. Okay, and when zinc is deficient in the diet, it may result in impaired growth and development, skin lesions and loss of appetite. Okay, next is which of the following will oscillate the fastest? So a large mass on a weak spring, a large mass on a stiff spring, a small mass on a stiff spring. So this is the one which will oscillate the fastest. Next, number 17. Which one of the following is or are the advantage of HPLC over the traditional LPLC, which is low pressure liquid chromatography? So what is the advantage of HPLC over LPLC? So advantage is, let's find out the correct option. Greater sensitivity and reusable columns, ideal for ionic species and larger molecules, sample recovery. Okay, so all of these points are correct for HPLC. That is why HPLC is preferred over LPLC, which is HPLC have greater sensitivity and the columns are reusable. The sample can be recovered and ideal for ionic species and larger molecules, for which LPLC cannot be used. Okay, so all of the above statements are correct. Next, in an agarose gel, DNA is moving from. Okay, so we already discussed in the previous questions that in gel electrophoresis, or be it SDS gel or be it SDS page, So what we saw that the DNA, okay, uh, okay, it moves from, okay, so we discussed in the last question that in both gel agarose gel electrophoresis, we saw that there is a movement of DNA towards the anode. Okay, so A option is correct. Next is number 19, fructose diphosphate accumulation would. Okay, so what will happen when fructose diphosphate will be accumulated in the body? It will stimulate glycolysis and it will inhibit gluconeogenesis. Now this is again a very important question, guys. What will happen when fructose will accumulate in the body? It will stimulate the glycolysis and it will inhibit gluconeogenesis. Okay. Next is, what is the cause of the genetic disease known as galactosemia? So what is the cause of galactosemia? That absence of galactose 1 phosphate uridyl transferase. Okay, this is the reason when galactosemia occurs. When there is the absence of galactose 1 phosphate uridyl transferase. Okay. So guys, let's begin with question number 1 of today's class. So it is like, which of the following is a non-standard amino acid? So non-standard amino acid is number C, which is hydroxyproline. Okay, cysteine, isoleucine and histidine all are standard amino acids. Next is proteins and macromolecules structures take on their higher order structures. How they take on the higher order structures? By self-assembly, with the help of molecular chaperones, with the help of precursor sequences that are removed from the final structure. So by all of these three processes, proteins and macromolecules, they take on the higher order. Okay, so all of the above will be the correct answer. Next is in ion exchange chromatography. In ion exchange chromatography, proteins are separated best based on their net charge. Okay, based on their net charge, proteins are separated in ion exchange chromatography. Next is an allosteric activator. An allosteric activator increases the binding affinity, decreases the binding affinity, stabilizes the R state of the protein, both A and C. So first of all, allosteric, when it is activator, it is increasing the binding affinity. So when it is increasing, it cannot be decreasing. It stabilizes the R state of the protein. Okay, so both A and C are the correct option. Next, number 5. What is a protein produced by host cells infected by viruses? So which protein is produced by host cell when the host is infected by the viruses? So it is interferon. So guys, this is also a very common and important question. Next is, the HIV virus infects primarily. So the HIV virus, we all know it infects the immune cells. Okay. Next is, the smooth endoplasmic reticulum is the site of. So it is the site of lipid synthesis. So number A, phospholipid synthesis will be the answer. Next is number 8. Number 8 is DNA helicase is used to. So DNA helicase, it is used to unwind the DNA double helix. It is used to break down the hydrogen bonds. Okay, we already discussed this. It is used to break down the hydrogen bonds between the base pairs. Okay, next question. Next is codon that specify the amino acid often differs in. So they often differs in the third base. Okay, the codon differs in the third base. Next is, which of the following is not necessary for protein synthesis to occur once transcription is completed? So once the transcription is completed, which one of the following is not necessary. So which one will be the correct answer, guys? The correct answer will be DNA. Okay. So DNA will be not necessary at that time. Now, degenerated codon differs mostly in. So it identifies the identities of their second base, the identities of their third base, the vowel position, both B and C. So the identity of the third base and the vowel position is where the degenerated codons differs. Okay, so remember, it's a third base and the vowel position is where the degenerated codon differs. Okay, so answer will be both B and C. Next is reverse transcription, transcriptase, it will be transcriptase from HIV does not. So reverse transcriptase from HIV does not. So remember, it does not have a 3' to 5' exonuclease activity. Okay, reverse transcriptase does not have, I'm writing it, 3' to 5' exonuclease activity. It does not have 3' to 5' exonuclease activity. Okay. Number 13, different DNA polymerases play distinct role in DNA replication and repair in both prokaryote and eukaryotic cells. All known DNA polymerase synthesize DNA only in the dash by the addition of dNTPs to a performed primer strand of DNA. So ultimately it's asking that in which direction DNA synthesis occurs. Okay, so remember, it's very simple. Yes, can you guess? It will be 5' to 3' direction. Okay, this is very easy. I'm very sure you could have done all that. Now, next is which one of the following amino acids interact with DNA or RNA backbone? Okay, so the amino acid lysine and arginine react with DNA and RNA backbone. Remember, in the DNA packaging we have done that. Okay, next is the lac-operon is translated into dash proteins. So lac-operon is translated into 3 proteins. If you remember ZYA, so 3 proteins have been produced. Number 16 is a human cDNA library. It contains what? So human cDNA library contains DNA for virtually all of the human proteins and vectors. Okay, it contains all of the DNA for virtually all of the human proteins and vectors. Okay, next is most of the enzymes of the citric acid cycle in a eukaryotic cell are located in. So where does the citric acid takes place? Citric acid cycle takes place in mitochondria. So where are these enzymes will be located? So these enzymes will be located in the mitochondrial matrix, which is the site where the TCA cycle will take place. Okay, which one of the following is involved in energy production mechanism? So generation of proton gradient across membrane, it produces energy. Transport of electrons on organic molecules, it also produces energy. Conversion of compounds with high energy to those of low energy. This also is involved in energy production. So all of these statements are involved in energy production. Number 19, a person with phenylketonuria is advised to not to consume which one of the following product. So this is a question which is repeated and this is an important one. They are advised not to take aspartame in their diet. Okay. Next is molar absorptivity. Absorptivity is a measure of the. So we already discussed, this is also a repeat question. It is amount of light absorbed per unit of concentration. Okay. This is the correct answer.